Welcome to The Explainer. Today, we're unpacking the fascinating and honestly kind of daunting biological journey of how cancer actually spreads through the body. It's this incredibly complex process of survival and navigation operating right under our noses, and understanding it, well, that's the absolute key to unlocking how we fight this disease.
Here's our roadmap for today. We're going to cover what is cancer, the threat of metastasis, the journey of spread, routes and destinations, and finally, the molecular toolkit. Let's get right into it.
Section 1. What is cancer? A cell division breakdown.
Normally, human cells follow a really strict life cycle, right? They grow, they multiply when the body needs them to, and they politely die off when they get old or damaged. But to start making sense of when this breaks down, we have to draw a hard line between benign lumps that just stay put and malignant tumors. Benign tumors generally don't invade nearby tissues, and if you remove them, they usually don't grow back. But cancerous tumors? Entirely different beasts. They invade local tissues and have this devastating biological drive to travel to distant places in the body to set up entirely new tumors.
These malignant cells basically go rogue. They completely ignore the body's normal stop-and-go signals. They won't stop dividing, they refuse to die, and they actively hide from our immune system, which is usually our main defense against damaged cells. On top of that, they'll even rewire how they get energy from nutrients just to fuel this massive growth spurt, all while racking up chaotic changes in their chromosomes, things like duplications and deletions.
Section 2. The threat of metastasis, a lethal systemic disease.
So, here's the crucial point. Metastasis is that specific biological event where malignant cells actually leave their primary site and navigate through the body to cause secondary tumors. And why does this matter so much? Because this process is literally the main cause of morbidity and mortality for cancer patients. A primary tumor can often just be surgically removed, but these new secondary metastases, they are incredibly stubborn and resist most of the treatments we currently have.
And obviously, this spread has very real human impacts, causing distinct physical symptoms depending on where these rogue cells decide to land. If they hit the lymph nodes, you get lymphadenopathy, basically abnormal swelling. If they spread to the lungs, it triggers severe coughing and dyspnea, which is a really intense shortness of breath. Liver spread can cause hepatosmegaly, an enlarged liver, and jaundice. Spread to the bones leads to agonizing pain and fractures, while brain metastases show up as neurological issues like severe headaches, vertigo, and even seizures.
Section 3. The journey of spread, escaping primary tumors.
Looking at those devastating symptoms, you kind of have to ask, how exactly do these microscopic cells pull off this great escape from the primary tumor in the first place? Well, it's a perilous itinerary. First, cells physically detach from the massive primary tumor. They migrate through the basal lamina and then undergo something called intravasation, which is just a fancy way of saying they penetrate a blood vessel. Once inside, they have to survive the incredibly harsh, turbulent environment of the bloodstream. If they make it, they undergo extravasation, squeezing back out of the vessel to adhere and multiply in a totally new organ.
And this entire pathway is actually broken down into 9 highly specific steps. Think of it almost like a biological escape room where the cell has to solve sequential physical challenges just to survive. It kicks off with neovascularization to get a blood supply, then invasion. Once they're in the transport system, they actually form embolisms, basically clustering together with platelets and immune cells to build a little protective shield for themselves. Finally, they arrest in a distant capillary bed, stick to the vessel wall, and squeeze out into the new environment to grow.
Section 4. Routes and destinations. The highways of spread.
Once a cell breaks out, it needs a biological highway system, and it basically has 3 primary transit routes to choose from. Route 1 is hitching a ride in the lymphatic system, invading the walls and riding to regional lymph nodes. Route 2 is diving directly into the bloodstream by punching through thin-walled capillaries, which honestly offer very little resistance. And Route 3 is direct extension, meaning the tumor just grows directly into open body spaces, like the pleural or peritoneal cavities. Now, this brings up a really fascinating biological mystery. If these cells are traveling everywhere in the blood and lymph, literally circulating throughout your entire body, why do they only seem to colonize very specific destinations?
It turns out metastasis has what we call organ specificity. Essentially, these cells have distinct preferences for where they want to unpack their bags. For example, cells from primary breast, prostate, or kidney cancers carry molecular passports that are heavily stamped for bone tissue. Colon cancer, on the other hand, is strongly drawn to the liver. It's also super interesting to note that some organs, like your skeletal muscle, your heart, and your skin, are almost never colonized by metastatic growth.
Section 5. The Molecular Toolkit. Tools for Escaping.
To pull off this grueling journey and land in exactly the right spot, the cancer cell needs a highly specialized microscopic toolkit. The absolute first tool it reaches for, angiogenesis. For a tumor to get bigger, it desperately needs adequate blood for oxygen and food. So, it literally forces the host's body to build new blood vessels directly to it by releasing angiogenic factors like vascular endothelial growth factor, or VEGF. This is brilliant, right? It secures an endless buffet of nutrients and paves a direct getaway highway for metastasis.
Next up, the cell hits natural tissue barriers, like basement membranes. To break through, it uses proteolytic enzymes as biological bolt cutters. Enzymes like urokinase and various cathepsins, specifically B and D, don't just act alone. They trigger this massive cascading chain reaction that literally dissolves the body's natural extracellular structures. They are actively carving a physical tunnel for the tumor cells to escape through.
Once it carves its way into the bloodstream, the cell whips out its first adhesion tool, integrins. These are membrane glycoproteins made of alpha and beta subunits, but you can just think of them as microscopic grappling hooks. They allow the traveling cancer cell to latch onto extracellular proteins and the walls of blood vessels. Without these, the cells would just be helplessly swept away by the sheer turbulent force of the blood current.
It also uses a tool called cadarins. These are calcium-dependent adhesion molecules that link directly to the cell's internal skeleton via proteins called catenins. Basically, they act as incredibly strong structural anchors. This allows the cancer cell to safely and securely lock onto other host cells it bumps into along its treacherous journey.
And we absolutely cannot forget about CD44. This is a family of cell surface proteins that acts almost exactly like a molecular GPS system. In healthy persons, CD44 guides our immune cells through the blood to where they need to go. But cancer cells hijack this exact mechanism. They use these CD44 receptors to bind to hyaluronate, which helps the rogue cell home in on the exact specialized tissues it wants to invade.
Finally, the cell's engine needs fuel, and that comes from motility factors, or motogens. These are unique peptides, like scatter factor or autocrine motility factor, that aggressively stimulate the physical movement of the cell. And here is the truly devious part. A lot of these motogens double as growth factors. That means they are simultaneously slamming the gas pedal for the cell to move and commanding it to rapidly divide.
So what should you take away from all this? Yes, the journey of metastasis is terrifyingly complex, but identifying every single piece of this biological toolkit is exactly how we fight back. By decoding how the roads are built with angiogenesis, how the fences are cut by proteases, and how the cells anchor themselves with integrins and CD44, modern science can eventually lock down these escape routes and save millions of lives. As we uncover each specialized tool these cells use, it leaves us with one provocative question. How close are we to finally building an inescapable biological prison for cancer?